without further ado, let me start. I'll talk about improving resource capture in nutrient and water limited environments. And it's a breeding approach based on root system modeling, genomic prediction, and on-farm selection, and other things. And my outline, I will touch on 4 different topics. The first 3 are summarizing a little bit my activities over the last 10 or more likely 3-5 years. And so there's a genetic component, genomic component. There will be a part about applied breeding in Madagascar. Then I'll switch to root modeling, and then in the end, I'll talk about upcoming research that is possibly touching upon what I may do in Bonn. So let me start with one of the highlights. It's a while ago, 10 years ago, but we cloned a gene that's called PISTL1 for pea starvation tolerance. And this gene was found based on some QTL mapping of the PAP1 locus. PAP1 stands for P uptake. And so through positional cloning, we found this protein kinase gene, and we could see that the gene is expressed at the very early stages of crown or nodal root development. And we could also, we know that it was very strongly upregulated under P deficiency. So it was a very good candidate gene in terms of the response to P. Here you see that it's very strongly expressed here at the base of the stem where the crown roots develop. And then overexpressing it in the variety that doesn't have the gene really enhances crown root development. And with more crown roots, it also enhances phosphorus uptake, of course. Now, one interesting part of this gene is that it's completely absent in most modern rice varieties developed for irrigated rice. And so it is not a question of having a different version of the allele, but the gene is absent, and actually a whole chunk of chromosome in that region is absent from these modern varieties. This is not usual. Anyway, after having gotten a handle on how to improve possibly phosphorus uptake, it was time to move to another trait, and I call it root efficiency, which I define as the P taken up per unit root size. So the first gene, the PISTL1 gene, increased root size. Now we make roots more efficient, hopefully. And a lot of these studies are based on Nerica 4, which is a popular upland rice variety in Africa, but that lacks adaptation to phosphorus deficient conditions. And the contrasting genotype we use is DG123. It's a gene bank accession from Bangladesh that we found in our screening experiments of gene bank accessions. And we can see here that the gene bank accession DJ has much larger roots, but then it has even higher phosphorus uptake. And that means that overall, the roots have even increased efficiency in uptake. And to see if this is linked to associations with mycorrhiza, for example, we have looked a bit at the colonization rate. There was no difference. We have looked at the expression of OSPT11. It's a phosphate transporter that is switched on specifically when the symbiosis with mycorrhiza is active. There's no difference there. And then we've confirmed that mycorrhiza are not responsible for this root efficiency effect. By growing plants in sterile soil, you can clearly see that the DJ, the dark bar, is far superior in efficiency compared to Nerica. So this was another encouraging result, that we have a donor in breeding that has a trait, an efficiency trait in terms of nutrient uptake. Now, a slightly different aspect of phosphate efficiency, and that is once the P is taken up, which can be done by bigger roots or more efficient roots, the question is how efficient is that P transferred into biomass or yield in the end. So this is PUE, the biomass produced per unit P. And it's difficult to phenotype, and not a lot of studies exist on PUE. And part of the difficulty is that PUE strongly decreases when P uptake increases. That has to do with the stress level plants experience. So the more stress they have, the more efficient they have to use the limited nutrient. So in phenotyping, it's very important to compare genotypes at an equal stress level. And we came up with a solution by growing them in small bottles, individual bottles with a known amount of fixed P that is easily accessible. So we can assume that all the P is taken up. And then we can measure the biomass produced, and we can move to genetic studies such as genome-wide association mapping, which we've done here. And we identified loci for POE on chromosomes 1 and 11. And very interestingly, DG123, the donor for root efficiency, is also a suitable donor for internal P utilization efficiency. That may indicate that it came from an area in Bangladesh, possibly northern Bangladesh, where P deficient soils predominate and were evolved under such conditions. So having such a donor would allow us to do breeding. But before I go to this applied breeding, I want to do one more, or touch on one more topic, and that is genomic prediction. I've already mentioned gene bank accessions, and my interest is to really find new variation, new traits that are sort of hidden in these gene banks and make them usable in plant breeding. But one problem with gene banks is that, for example, the IRI gene bank has more than 100,000 accessions. Where do we start to test gene bank accessions? And genomic prediction is one way to target this approach and be rather efficient. So we use, in this genomic prediction, we use genome information, as many markers as we can have, or features, genetic genomic features that we have. And then we relate that to phenotypic data of the gene bank accessions that we test, and through some model, we can predict the likely phenotype of a gene bank accession. Now, IRI produced a resource that I have used a lot, and they have provided the genome information after sequencing 3,000 of their 100,000 rice accessions. And now, I mean, even a field experiment with 3,000 accessions is a bit too large for my taste, so I'd rather concentrate on a smaller number. So what we have done is take 300 of these accessions, so 10%. We've grown them in Madagascar, we phenotyped them, and then we use the phenotype data in this sort of model to predict the phenotypes of the remaining 2,300, so of all the 3,000. And then we can further test whether our predictions are correct or not. And we've done that for traits such as yield under phosphorus deficiency. We've done that for zinc biofortification, and we will hopefully do that for root traits sometime soon. And so here's how the flow is for this genomic prediction. So in the first year, we have the phenotypic data of the 300 tested ones. We then use the model to predict the performance of all 3,000. And for example, we can then go ahead. So these are the 3,000 predicted performances. And then we can select the top, the bottom, and the middle end, for example, and test them, import them to Madagascar, test them under similar conditions. And this is what you see here. So actually the top end that was in red here performed much better than the middle or the bottom end. So while of course it does not work perfectly, but we are rather happy with the accuracy of prediction of about R0.5 in this case. And then we can go about and repeat this a second cycle and see whether we can further improve our prediction accuracy, and that you can see here. So this is a nice academic exercise, but we started this to actually identify useful accessions from the gene bank, and this was actually possible. So we particularly like this one here, accession 1103, which combines relative high yield with earliness. And we use that as a donor in our breeding, and so we have a breeding population now that we test in Madagascar. Now let me switch to the second topic, our applied rice breeding activities. Madagascar is a country where P deficiency abounds, so it's a perfect place for me to do research and try to achieve some progress with applied research as well. And as you can see here, very often we have fields where conditions are like this, where you see a very clear response to fertilizer applied. And farmers typically do not have the financial means to apply fertilizer. That is unfortunately reality here. So in order to achieve something with our breeding work, I made a decision early on that we do all our selection and all our experiments in farmers' field under these low input conditions. And that is what you can see here. So rather than going to a research station where conditions are typically quite a bit better than in average in the country, we focus on these farmers and do our experiment under low input conditions there. And in this case, you see we've done GWAS and genomic prediction here. This was a QTL mapping experiment. Here our breeding trials. And the breeding trials in particular now have borne fruit. So we have concentrated in our variety development on 2 breeding populations. The first one is based on this PAP1 and PISTL1 gene that I introduced in my first slide. We've done marker-assisted selection for PAP1 to bring it into the IR64 background. IR64 is globally the most successful rice variety, but it has low adaptation to this low P-deficient conditions, and it was never adopted in Madagascar. But otherwise, it's a really very good variety with many positive attributes. So we're trying to improve it by bringing the PAP1 locus into IR64. And the second one is using the donor DG123 in a cross with IR64. We've done all our selections in farmers' fields. And because we combined, so we did part of the work in Japan already. We brought breeding lines from crosses made in Japan to Madagascar, and we combined it with off-season generation. So as a result, we were rather fast in producing advanced breeding lines that could enter the 2-year variety registration process. And this involves farmer participatory evaluation. So we often get feedback from the farmers that we work with. That also involves sensory evaluations to make sure that the taste and other cooking qualities is acceptable. And of course, we have to worry about grain yield, which we improved by about 15 to 20% in this case. And in the end of last year, we were then actually able to release 2 new varieties. They're called Fivari 32 and 84. Fivari is the name of our big project. And they were handed over to Malagasy stakeholders in November last year. And we are now in the process of producing seeds so it's time for me to go home. Sorry, guys. And this kind of activity increases the visibility of a project quite dramatically. And the guy that you see here, he is the president of Madagascar, looking at our variety information pamphlet and being apparently quite happy about what we've done. This breeding activity has been, the first part has come to a conclusion, but we have several other pipelines that are still ongoing. So we are trying to improve upland rice, and 3 advanced breeding lines just completed the first year of the variety release process. Then we have some lowland rice populations that we try to adapt to water scarcity in Madagascar, and we work on zinc biofortification, so trying to increase the zinc content in the rice grain. And since these are really quite advanced activities, I definitely hope to continue these activities in the future. So now let me switch to something completely different, modeling of roots and rhizosphere processes. And why would a geneticist and a breeder like me be interested in modeling? And that is that because on these highly phosphorus fixing soils, like undersoils in Japan or oxysoils in Madagascar, the P uptake processes are very poorly understood. And I think breeding and selection would really benefit from an improved mechanistic understanding of the traits that drive higher phosphorus uptake. So I had a PhD student that worked on developing a model for DG123. Before I go into details, I want to briefly mention some specificities about rice roots. So rice has a much finer root system than most other cereals. It has, here's the primary root, and of the primary root are the lateral roots. That's nothing special yet. But then all root types bear a second class of lateral roots. They're the S-type laterals. Here you can actually see one sticking out, but they are very fine and almost invisible. Here you can see on one crown root, you have the L-types and then the small S-type laterals. They are really unique to rice. They have a fixed length of about 1 centimeter and a very narrow diameter. And very interestingly, for their small size, they still bear root hairs. So here we have these S-type laterals with root hairs. So it's a microgram. This was something that my former postdoc, Josephine Kant, who's now newly discovered a few years ago when she was here. Now, we have modeled all these root structures in our model, and let me see. While I run the model here in the background, you can see there's the root growth modeled over 30-some days with crown roots developing first. You can see the main L-type lateral roots developing here. S-types develop on all kind of roots, but I guess they are visible if you look very closely, and then root hairs are actually invisible. Here you have another set of crown roots developing from tillers, and the model was calibrated against root scanning data and roots that we dug up and measured in pot experiment and field experiments. And here is the model prediction versus the measured root data, and you can see that we quite closely match that data. And here is the number of nodal roots or crown roots developing. And then back here is the estimation of P uptake. And we can predict P uptake very well until about day 35. And then at a later stage, we still underestimate P uptake. Anyway, one, I think, very interesting result from this modeling was a cost-benefit analysis we done for different root classes. So the cost for us is the P invested in root biomass, because we do our modeling under P limiting conditions. So P is really the growth limiting factor, and therefore the cost we have to consider mostly. Benefits would then be the P taken up by a class of root. And so the model simulation showed that, first of all, most of the P uptake came from nodal roots, despite the fact that hairs and fine structures are much, much longer than the roots themselves, the nodal roots themselves. But costs in terms of P invested are very unfavorable for nodal roots. That's because of the large diameter. They're much heavier than these fine structures, and therefore the total cost invested is much, much higher. And now we can actually look at the efficiency in terms of P uptake, in terms of length. Okay, the nodal roots are still good, but in terms of weight, the nodal roots have a very strong disadvantage over fine structures. And if we relate the benefit in terms of P taken up versus the cost, we clearly see that the root hairs and the S-type roots have a very, very strong advantage over the nodal roots. And we can estimate cost recovery in terms of days. And this is actually something relatively new and interesting, that this cost recovery is very rapid for fine root structures like hairs and S-types, but it's very slow for crown roots. And that may already explain one observation that we've seen in the field many times, and that is that crown root number is the trait that is very, very strongly reduced under P deficiency in rice. And it makes sense to us now that the crown roots are such a big cost to a plant that it would do well to reduce it, unless it can maintain crown root development by higher P uptake. Now, a second model that addresses solubilization in the rhizosphere. The problem we have with almost all plant growth models or phosphorus uptake models is that they strongly underestimate the actual uptake achieved by rice on these strongly P-sorbing soils. So if we measure the P concentration available in these soils and use it as an input, we underestimate P uptake by sometimes 90%. Christian Kuppe, who is in Jülich, working with Johannes Postma, he developed a new model that allows for solubilization of P as a result of pH increases. And the pH increases because we assume that plants take up nitrate in excess of ammonia, and then to balance the negative charge, the pH will increase. And then the important point is that the pH gradient moves much further in the soil than the solubilized P can diffuse back to the root, and that is shown here. So we have the pH gradient extending, but the diffusion of P back to the root where the pH gradient originated is really limited. And so we have a P depletion zone here, but a zone where P is solubilized that is much larger, but that the plant, the original root, cannot access. And now the trick is that there's some synergism between this pH change and the solubilization and having these S-type lateral roots, particularly if these have hairs, because these small root structures that take up much less nitrate and affect the pH to a much lower extent as a main root, but these roots extend into these solubilization zones and can mop up the P that was solubilized because of the action of the main root. So for the first time, this model can actually explain the P uptake we observe from upland rice. And as a result, we see that the rice root fine structures play an even greater role than we had originally thought. All right, so now that brings me to already just a bit of an outlook of what I'd be interested in doing when I'm in Bonn over the next years. One question is to solve what is driving phosphorus solubilization in these soils, and do genotypic differences exist for these traits? And we have modeled everything in response to nitrate uptake, but it's also very likely that other exudates exist that solubilize soil-bound phosphorus directly, or that these exudates interact with microorganisms that solubilize P. We don't know which of these processes is more important, but we have a project to at least address these questions, and it's a project that just started last year and will still continue for almost 2.5 more years, called rhizobreeding, together with partners at SALF and BOKO in Vienna and in Jerkes, of course. And then there's a continuing collaboration with Jülich on the modeling aspects of these, and actually on some rhizosphere measurements, and with Cranfield University. It's an old partner of mine that is involved in similar kind of research. Another project that I'm actually has just started, and I'm rather excited about it, because the resource that we received from the partner Rothamsted is such a unique, fantastic resource. So we have received seedling root images like these. Here there's 2 rice genotypes in 1 plate. And we have images of 600 of these rice gene bank accessions, all fully sequenced. So it's a very unique resource that will allow us to identify traits for different root traits. And you see that there's clear differences in terms of the maximum length on the branching density, the root to shoot ratio, and many other traits. So this, I think, could play a rather big role in my research, and it involves phenotyping all these different root traits, which could include, as I said, root to shoot ratio as well, and densities and length. And based on the phenotype, one can apply several genetic approaches to identify candidate genes. Again, to do genomic prediction to predict even more extreme phenotypes among the gene bank accessions, to predict idiotypes, for example. And if we can define and select idiotypes based on this data, we can do hypothesis testing in soil. Do these contrasting idiotypes really affect performance as we have predicted them from our model or not? And this would allow interaction with modeling groups and so forth. A big question in all of these is how to extract these phenotypes. We have more than 1,000 images of these, and you can imagine that it's not a small feat to turn these images into usable data that can go into these processes. But in the end, what I hope is to find donors, markers, new alleles that can be used in breeding. And I think this is certainly one activity where there could be quite a few interactions with PhenoRob in different components.